Thank you. Hello, I'm Sangjun Jeong, a PhD student under Dr. Niu and Dr. Jand. Thank you for giving me the honor to introduce Dr. van Iersel today. Dr. Marc van Iersel received his PhD from the University of Arkansas in 1994. He has been with Horticulture Department of University of Georgia since 1995, where he now holds the Dooley Professorship. His research focuses on cost-effective supplemental lighting technologies in greenhouses and vertical farms. He is the director of Project LAMP, which stands for Lighting Approaches to Maximize Profits. It's a $5 million US-based research project that brings together plant scientists, engineers, and economists to develop profitable supplemental lighting strategies. In 2017, he co-founded a lighting technology company called Candidates to help bring novel lighting strategies to the greenhouse industry. Dr. van Iersel has published over 140 scientific papers and has given invited lectures about his research around the world, including in Italy, Spain, Taiwan, Kenya, Canada, Chile, and Brazil. I am very pleased to introduce him and excited to hear what he has to say today. Let's all welcome Dr. van Iersel. Thank you.
Thank you, Sangjun. I think it's especially appropriate that you get to introduce me today, since not only did you receive your master's degree under the guidance of my former PhD student, Jong-Yun Kim, you're now pursuing your PhD degree being co-advised by another former student of mine. So we have a lot of heritage in common. So thank you for that introduction.
Yes, thank you, thank you.
It's a small world sometimes. And thank you to the department for inviting me to talk to you today. I'm sure we all wish that this could be in person. So what I will be talking about today is smart lighting strategies for controlled environment agriculture and how we go about developing those lighting strategies in my lab. Before going into the nitty-gritty of the science, let's start with a quick introduction into controlled environment agriculture. In my mind, we can divide controlled environment agriculture into two different categories, and that includes greenhouse production. And so in greenhouses, it is often necessary to provide supplemental light during the darker parts of the year to assure the production of high quality. Sorry, my cat is now messing with my computer. It is necessary to provide supplemental light for high quality production during the darker parts of the year. And that supplemental lighting can be expensive. So in greenhouses, the cost of providing that supplemental light can be about 15 to 30% of total operating costs. So the other component of controlled environment agriculture that has gotten a lot of media attention recently is vertical farms. And so vertical farms refers to the production of often leafy greens, maybe herbs, in a completely indoor setting, often in a vertical manner, as you can see right here. And this has become really popular. It has gotten a lot of media attention, but was still a lot of questions about the actual profitability. An exciting story that came out very recently is that the first vertical farm is now going public. And so Aero Farms, which was founded by someone I know quite well, is currently valued at $1.2 billion with a B dollars. So this is getting to be a very big business. And so in vertical farms, lighting can account for about 30 to 50% of total operating costs. So it's a big deal. So that explains why the research that we do in my lab focuses on lighting. It is the most expensive, and especially in greenhouses, the least or most poorly controlled environmental factor. And so good lighting management, good strategies to provide that supplemental light can easily make the difference between a greenhouse facility making a profit versus losing money. But I have a third reason why I wanted to work on lighting, and that third reason is entirely selfish. Because lighting was just a great topic to combine my interest in plant physiology, and I've long had an interest in photosynthesis. Combine that with LEDs, instrumentation, automation. This has turned out to be a great field where these different things can be combined, and that is something that I have long tried to do in much of my my research. So, before getting started on the actual research, I want to briefly talk about the philosophy behind how I do my research, especially something for the graduate students to think about. So what I really want to do is start out with understanding the physiology that is going to drive plant responses, whether that is related to lighting or anything else that we may be studying. And based on my understanding of that physiology, then develop testable hypotheses. And once you have a good hypothesis, you can design an experiment to test that hypothesis and hopefully then carefully execute that experiment. And then one thing that I also want to really emphasize is that I probably spend more time after the study analyzing my data than most people. And that is something that I do very deliberately, because I think it is really important to make sure that you get as much information out of the data that you collected as you possibly can, right? You spend a lot of time doing a study, so you might as well spend a lot of time trying to understand your data as well. And so that is something that I really try to imprint on my graduate students. So when it comes to lighting, there is one really obvious problem, and that is that if you are growing you have small plants and you're providing lighting, as you can see here with these seedlings, is that only a small fraction of the light that you're providing is actually going to hit one of the leaves on these plants. So we have poor light capture here, and we can clearly increase the efficiency of supplemental lighting if we can make sure that more of the light we provide is actually going to hit a leaf, so it at least has a chance of contributing to the growth of that plant. And there is also an obvious solution, or at least partial solution to that problem, and that is manipulating the light spectrum to manipulate the morphology of the plants, and specifically playing around with far red light, which induces phytochrome responses in plants and can induce a shade response. And so phytochrome is a protein complex. There's actually a complex between a protein and a light receptor that occurs on plants. And it can exist in two forms that absorb very differently in the far red part of the spectrum right here and the red part of the spectrum. And phytochrome is used by plants to detect whether or not they're shaded by other plants. So when we look at the light spectrum in full sunlight, you can see that the intensities of red light right here are roughly the same as far red light. But when you measure the light spectrum underneath a plant canopy, now we can see that there is a lot more far red present here as compared to red light. And so that shift in the amount of far red light compared to red light is something that can be detected by phytochrome, and that can then induce shade responses. And this has been studied for a long, long time, including with regard to lighting in horticulture. And one example of that is work that was done by William Meng and Eric Runkle at Michigan State. So here we have two trays with the same cultivar of lettuce, despite the fact that they look very differently. But the plants on the left that you can see right here were grown without far red, and the plants on the right were grown with far red light. And you can see that the plants on the right make much bigger leaves. They have a much bigger canopy. And because of that, they can then, of course, capture more light energy, and that light energy can then be used to increase growth. And so there have been quite a few studies on this topic already, and specifically with a focus on lettuce. So before we decided to start working on this topic in my lab as well, we needed to figure out, can we actually contribute something to this topic that is new? Because if we cannot contribute new information, then there was little point in us working on this topic. And so one thing that we didn't really discover in our lab, but we rediscovered it, I should say, is that far red light has photosynthetic activity. This was first discovered in the 1950s, I believe, but it seemed to have been largely forgotten. And even in the people who were working with LEDs and far red light, there seemed to be little awareness, actually, of the photosynthetic activity of far red light. So this work was done in my lab by Shuyang Zhen, who is, of course, now part of your faculty. She continued that research at Utah State. So we now have very convincingly proven that the photosynthetic activity of far red light is important. But also, when I looked at the literature, there were some questions that remained unanswered. And that is, how much far red should actually be used in a light fixture? And is far red more effective in increasing growth than adding the same amount of photosynthetically active radiation? And so that refers to light with wavelengths from 400 to 700 nanometers. And so that also was a question that really hadn't been answered very well. From a practical perspective, if we want to provide far red light in plant factories, what that typically would mean would that we would remove some of the LEDs that are providing photosynthetically active radiation and replace them with far red photons or far red LEDs. The question is, do we get better growth when we replace photosynthetically active radiation with far red photons? And then we had another very practical reason to do this research. A new graduate student had somewhat unexpectedly shown up in my lab, and I needed to figure out something for him to do. In addition to that new graduate student, we also had just gotten a new imaging system, and we needed to give that imaging system a try. And so we designed what we initially thought was going to be a relatively simple and not overly innovative study, just to see how well this new imaging equipment that we had received actually worked and what we could do with it. So the imaging system that you see here, the top view phenotyping system, is actually a multispectral imaging system. But what we were most interested in was the ability of this imaging system to take monochrome or black and white pictures of chlorophyll that is actually given off by plants. And for those of you who are not aware of chlorophyll fluorescence, every single plant, when it is exposed to light, is actually fluorescing. So it is giving off a small amount of light. And so typically about 1% of the light that hits the plant is re-emitted by that plant in the form of fluorescence. And it turns out that there is a really simple way to take pictures of that fluorescence. So here you can see a lettuce seedling. It's in a 4-inch pot. And so you see the two cotyledons right here. And in the background, unfortunately, you see a little bit of algae that is growing on the top of the potting soil that we were using. It turns out that algae also fluoresces very well. And so we had to learn how to effectively suppress algae without hurting the plants. But so if you take pictures of these plants over time, you get images that look like this. So this is the same plant. So it provides a really simple way to monitor the growth of plants over time. And it's non-destructive, which is really nice. And so this allows us to really see how differences in plant growth occur in response to different treatments, and in this particular case, in response to far red light. And this seemed like a good match to do a study with far red light, because the response that we were expecting to get from far red light was increased leaf elongation. And as you can imagine, with these images, quantifying that change in leaf elongation in response to far red light is something that would be pretty straightforward. And we have now developed a fully automated way to actually analyze these images. And basically all that our software does is now count the number of relatively light pixels, and that is a direct measure of what we call the projected canopy size. So the student that got to do the first study using this imaging system was Dr. Yves Legendre. He graduated last fall, and the paper that he wrote on this topic was published about 2 months ago, so earlier this year. And what we wanted to do is really understand both the morphology and physiology that is affected by far red light. So for the first study that he did, we started off with a really simple hypothesis. And that was that far red light will increase light capture because it increases leaf elongation and therefore growth. That was, to be honest, kind of a no-brainer. We would have been shocked if that had not been the case. But what we were also expecting was that far red light will increase growth more than an equal amount of photosynthetic active radiation. And if that would truly be the case, that would be a clear indication that in grow lights, it is actually worthwhile to replace some of the more typically used LEDs that provide 400 to 700 nanometer light with far red LEDs. So in the first of the 2 studies that Yves did on this topic, he created a far red gradient inside of a growth chamber. And then he grew green salad ball lettuce. He harvested some plants when they started touching each other. Basically, at that point, we harvested half of the plants in a zigzag pattern to give the remaining plants more room to keep growing. And the far red gradient that he created in the growth chamber exposed plants to far red intensities of roughly 5 to 30 micromoles per square meter per second. And all of the plants got roughly the same amount of photosynthetic photon flux density. So that is 400 to 700 nanometers. So the one variable in the study really was the amount of far red light that these plants received. So in this graph, you can see data from the 2 different harvests that Yves did. So the gray circles in this graph and the following graphs was from the first harvest, and the red triangles is from the second harvest. And as we expected, we found that increasing the amount of far red light, so going from left to right, increased leaf width. So we have expansion in that direction, but it also increased leaf length. So with far red, we clearly were getting larger leaves than with low amounts of far red. That wasn't overly surprising. And then when we looked at the relationships between leaf length on the left right here and leaf width on the right with projected canopy size, we see that for both of the 2 harvests that Yves did, as the leaves get bigger, projected canopy size increases. And yes, I realize that seems rather obvious, but it was really nice to see these strong correlations. So bigger leaves, more canopy, and more ability of the plant to capture light to support its growth. And we've also looked at the relationships between leaf length and the total leaf area of the plant. So this is not projected canopy size, but this is actually all of the leaves from the plant harvested and then measured and added up together. But once again, we see that if you have larger leaves, not surprisingly, the leaf area increases. So all of this was, I would say, pretty obvious. But when we were thinking about these data, and this is something that, to be honest, we hadn't thought about when we started the study, but then we realized we can use these data to actually calculate, or at least estimate, the total amount of light that these plants receive over the course of the growing cycle. Because when we plot the projected canopy size that we measured, so that's the symbols in this graph, over the course of the study, we can then fit a regression equation to those data, and that regression equation always fits nearly perfectly. And then using this regression equation that you see here, we can calculate the projected canopy size on each day throughout the study. And if we know how large the plants are on a specific day, and we had measured the light that each plant received, then we can multiply the light intensity in micromoles per square meters per day by the projected canopy size in square meters per plant, and we can calculate for each day how much light each of these plants get, and then we add that up over the course of the whole growing cycle, and we can calculate what we call the cumulative incident light that the plants received. So what I want to emphasize here, this is light that actually hit the plants. There are many other studies where they talk about incident light, but that typically then refers to light in the growing area, but that light doesn't necessarily actually reach a plant. So that is something that we were able to do because we had these projected canopy size measurements. So here we have the incident light in moles per plant on the x-axis, and in these two graphs on the y-axis we have projected canopy size at the two different harvests and plant leaf area at the two different harvests. And not surprisingly, we see that as plants have a larger canopy, either based on projected canopy size or total leaf area, they receive more light. That too was of course to be expected. And then we compared the total amount of incident light to the dry weight of the plants. And what we saw there was that the total amount of incident light is very well correlated with, sorry, cat is back again. It's very well correlated with the dry weight of the plants. And basically what this really emphasizes is the fact that light interception by a plant is really critical for plant growth and the accumulation of dry weight. What you probably didn't notice in the last few graphs, as I was describing the data, that I no longer showed actually the treatments that we started out with, but we wanted to look at the effects of far red light. So if we go back to far red light on the x-axis right here, what we see is that when far red light increases, so we go from left to right, the total amount of incident light increases because far red results in larger canopies and thus more light interception. And because we get a larger amount of incident light with higher levels of far red light, we also see that the dry weight of the plants increases with increasing far red. So this is what we had expected. And then when we looked at these data, one thing that was interesting here is that we increase, when we increase far red from about 5 micromoles per square meter per second to 20 micromoles per meter per second. So that's a 15 micromole per square meter per second increase. And that is actually a little bit less than 10% of the total amount that these plants received. Remember that they got 200 micromoles of 400 to 700 nanometers light. So an increase in the amount of light by less than 10% actually increased dry weight by 33%. So that was actually pretty exciting because we got a disproportional increase in growth in response to providing that far red light. So from this study, we drew two conclusions. The first one was, well, we were really happy with the way our imaging system worked. The second conclusion was more related to the actual hypothesis, and that is that far red light increases leaf elongation and therefore canopy size. That increased canopy size increases the total amount of light that reaches the canopy and therefore growth. But one question that we didn't have a very definitive answer to was, is far red light actually more effective than photosynthetically active radiation? So to more specifically address that second question, Reeve did a second study where he had a little bit different setup. Instead of having a far-red gradient inside of the growth chamber, we now created two gradients in the growth chamber where we had most of our white LEDs on the right side of the growth chamber right here. So we had high photosynthetic photon flux densities here and lower photosynthetic photon flux densities on the left. And then we placed our far red LEDs near the back of the growth chamber. So we got a perpendicular gradient with high far red from the back of the growth chamber towards the front of the growth chamber. So this gave us many different combinations of far red with photosynthetic photon flux density. Reeve collected a lot of the same data in this study as he did in the first study, but I'll skip over most of those and just get to the most important parts of it. And that is that specifically one of the things that he did was he did a multiple regression analysis where he looked at plant responses, and in this case it's leaf length, as a function of far red, as well as a function of photosynthetic photon flux density. And so what this graph shows here is that as the far red intensity increases, we get an increase in leaf length, very similar to the first study. And interestingly, as the photosynthetic photon flux increases, we actually get a decrease in leaf length. So higher light intensities in the 400 to 700 nanometer part of the spectrum decreased the length of the leaves. And when we did the same kind of analysis for leaf width, we see that far red light increases the width of the leaves, but 400 to 700 nanometer light actually had no effect on leaf width. So overall, what we saw here was that far red increased leaf size, while PPFD decreased leaf length, had no effect on leaf width, and just overall decreased the size of the leaf. Similar to the first study that Reeve did, he calculated the incident light that these plants received over the entire growing cycle. So once again, in response to different levels of far red light and different photosynthetic photon flux densities. And we see that increasing far red and increasing photosynthetic photon flux density both increase the total amount of incident light. But because we did this multiple regression here, from the slope of this plane, it actually has two slopes, one in the x direction and one in the y direction, we could compare how effective far red photons were compared to PPFD photons in increasing incident light. And when we did that, we actually found that far red photons were 183% more effective at increasing incident light than 400 to 700 nanometer photons. So the effect that far red light has on the size of the canopy is just really important in allowing these plants to capture light. And so I think this is probably the first time that I saw a clear quantification of the relative efficiency of far red versus PPFD with regard to the ability of plants to capture light. And then we did the same kind of graph for dry weight, where once again you see that increasing dry weight and increasing the photosynthetic photon flux density, they both increase dry weight. But once again, what we found here is that far red photons are much more effective at increasing dry weight than photosynthetic photon flux density. So here, 162% more effective, which basically means roughly 2.5 times as effective. So a very big difference. I do want to point out that that conclusion is only valid for this particular range of far red compared combined with this range of photosynthetic photon flux density. We need to be careful to not extrapolate from this. And then as we were doing these analyses and we were thinking about the dry weight data, we actually finally realized there was another really interesting parameter we can calculate from these data, and that is the light use efficiency. Because we know exactly how much light these plants receive over the course of their growing cycle. We, of course, measure the biomass of, in this case, only the shoots at the end of the study. And so when we divide shoot biomass by the total amount of incident light, we get the light use efficiency, which tells us the grams of biomass produced per mole of photons reaching the plant. And so when we look at the light use efficiency, and here we calculated incident light based only on 400 to 700 nanometer photons. When we plot that against far red light intensity, we see that this light use efficiency increases with increasing amount far red, which suggests that per mole of incident light, these plants are able to produce more biomass if they get more far red light. But this is a little bit deceiving because here we calculated the light use efficiency based on photosynthetic photon flux density. And remember that I've been trying to emphasize that far red light in reality is also photosynthetically active. So if we recalculate the light use efficiency now based on the total photon flux that includes the far red light, then we don't see any effect on light use efficiency, which basically means that regardless of how much far red light we added, the plants always used the incident light with equal efficiency to produce biomass. So here you can see some of the plants from the more extreme treatments with low far red and low ppfd at the top, to high far red and high ppfd at the bottom, and it gives you kind of an idea of the images that we get from our imaging system, and then the kind of information that we can derive from that. So from the second study, we were able to draw two conclusions that are, I think, important. And the first one is that far red light increased total incident light and growth more than an equal amount of photosynthetically active radiation. And so because of that, we concluded that for sole source lighting, so vertical farms, indoor plant production, light fixtures really should contain far red LEDs because they help drive growth of at least this type of lettuce a lot more efficiently than the standard white or red and blue LEDs that are more commonly used right now. Another advantage of including far red LEDs is that far red lights have a high efficacy. And basically what that means is that they're more energy efficient than other colors of LEDs. So you can get the same photon flux from far red LEDs with less energy. So it would have benefits from an energy efficiency perspective as well. Nonetheless, far red LEDs are rarely included in grow lights, which raises the obvious question, why not? Well, the reason for that is largely regulatory, unfortunately. So the efficacy of a light fixture is defined as the amount of photosynthetically active radiation that light fixture produces divided by the power the light fixture consumes. And there is an agency in the United States, the Design Lights Consortium, that certifies light fixtures based on their efficacy. And this certification is really important for lighting companies because that certification is used by utility companies to provide incentives or rebates for more energy efficient lighting systems. And so if lighting companies were to develop a better light that includes far red, unfortunately this efficacy measure would go down because the far red light doesn't count towards this definition of photosynthetically active radiation. And so because of that, far red unfortunately is still rarely used in light fixtures. And so that raises the question, shouldn't PAR be redefined? And so right now, that definition of PAR is based on a publication by the American Standards of Agricultural and Biological, the American Society of Agricultural and Biological Engineers. And that society has defined photosynthetically active radiation as light or photons from 400 to 700 nanometers. My question is, why in the world do we allow engineers to define this physiological measure? But it is really difficult to get engineers to change their mind regarding this. But I'm pleased to say that Shu Yang, Bruce Bugby, and I are now collaborating on really trying very hard to get this definition changed so that far red LEDs can actually be incorporated into light fixtures. With that, I'm going to shift gears and talk about a study that was done by my technician and PhD student, T.C. Jayalav. And it was based on a very simple observation that we had made in my lab. And that observation was simply that when we grow both Mizuna, an Asian green with great flavor, when we grow that under the same conditions as lettuce, then Mizuna grows much faster than lettuce does. And so that just got us curious as to why can Mizuna grow so fast compared to lettuce? Because ultimately, having crops that grow really fast in controlled environment agriculture can result in more crops per year, in principle, lower the production cost, and potentially increase the profitability of these operations. And so in addition to simply comparing Mizuna and lettuce, we were interested in also looking at how this was affected by the photosynthetic photon flux density. So this study was done inside of a walk-in growth chamber where we grew the plants under 6 different photosynthetic photon flux densities listed right here, resulting in daily light integrals. So the total amount of light these plants received over the course of a day, ranging from about 3 to 24.5. And in this study, TC measured all of the different properties, physiological and morphological, of these plants that we could think of that might help explain the difference in growth between these two crops. So I'll start off by giving you an idea of what these plants look like. So here we have the lettuce. Big shock, lettuce actually doesn't like very low light levels. And as you provide more light, lettuce grows more. Not really a big shock. That was kind of obvious that that would happen. When we compare that to Mizuna, we generally speaking also see that Mizuna does very poorly under low light and tends to get bigger as light levels increase, although some of these pictures here are a little bit deceiving. I don't think that the 200 and 275 micromole per square meter per second treatments really resulted in better growth than the higher light levels. So just like Reef had done before, TC took lots of images of these plants to get the projected canopy size. And so this graph is going to show the data from all of the treatments, but to get you oriented, I start out with one treatment right here where we have 50 micromoles of PPFD. And so this graph shows the projected canopy size on the y-axis over the course of the growing cycle, with the first 8 days blown up right here in this insert, because otherwise you can't see any of the differences between the two species. So when we add all of the other treatments to this graph, what you can see is that especially at higher light levels, Mizuna develops a much bigger canopy than lettuce does. And those differences actually start appearing pretty early on during that growing cycle. And one simple reason for that, actually 2 simple reasons, is that Mizuna germinates faster. Sometimes we can see it germinate the same day that we plant it. And Mizuna has larger cotyledons than lettuce does. So it has a head start because of those 2 different reasons. So this certainly suggested that the larger canopy that Mizuna develops is going to help it grow faster because there will be more incident light. But what also matters is how much of the light that actually hits the canopy is going to be absorbed by the canopy. And so to look at that, sorry, I'm getting ahead of myself. TC calculated the total amount of incident light over the course of the growing cycle for the two species. At very low ppfds, that was pretty similar, but at higher ppfds, Mizuna had higher incident light than lettuce, despite the growing cycle for Mizuna actually being a day shorter than it was for lettuce. So Mizuna has higher incident light, but it also needs to be able to absorb that light to be able to use it for growth. And so we looked at the chlorophyll content index, because higher levels of chlorophyll will help plants to absorb the photons that strike the leaves more efficiently. And what we saw was that as the ppfd increases, the chlorophyll content in both species increases, but the chlorophyll content index is much higher in Mizuna than in lettuce, and the increase with increasing light is greater in Mizuna than in lettuce. So what this suggests is that Mizuna is better able to absorb the photons that hit the canopy. So that then got us to the next question is, once these photons are absorbed, how efficiently are they used for photosynthesis? And to look at that, we used chlorophyll fluorescence measurements to determine the quantum yield of photosystem 2, which we have on the y-axis here. And so what that indicates is the fraction of photons that are absorbed by the leaf that are actually used in the light reactions of photosynthesis. And what we see is that this decreases for both species with increasing light levels, but Mizuna consistently has a higher quantum yield than lettuce, meaning that it can use absorbed photons more efficiently than lettuce. And then when we look at photosynthetic rates, which would be the next step, we see that especially at higher light levels, Mizuna tends to have a higher photosynthetic rate than lettuce does. So that is very consistent with what we saw regarding quantum yield. Like Reeve did before, TC then calculated the light use efficiency, and we see that very consistently, Mizuna has a substantially higher light use efficiency, especially at the lower light levels, than lettuce does. So what this indicates is that per unit of light that actually strikes the canopy of these two different species, Mizuna is able to produce quite a bit more biomass than lettuce can. And so based on all of this information, it then shouldn't come as a big surprise that at higher light levels, Mizuna accumulates a lot more dry weight or biomass than lettuce does. So from the study, what we were able to do is figure out why Mizuna grows so much faster than lettuce, and that is because it germinates faster. It has larger cotyledons, giving it a larger canopy very early on in development. It has more chlorophyll, allowing it to absorb incident light better. And because of the larger canopy, there is more light interception that results ultimately in a higher, well, also a higher photosynthetic rate, and Mizuna has a higher light use efficiency. So it has many advantages with regard to growth when you compare Mizuna to lettuce. So why does this actually matter? Well, we think that this is actually a very elegant way to screen for rapidly growing crops. And if you want to screen for differences in crop growth, either among germplasm or based on different treatments, you don't have to measure everything that we measured in this study. All you need to do is measure projected canopy size over time, which allows you to look at the morphology of the plants, and then you can calculate light use efficiency, which is a measure of the physiology of the plant. So it's a very simple way to get an idea of the reasons for growth differences among different crops or plants grown under different conditions. The third study that I'm going to talk about, this will be really, really short, I promise you. We wanted to answer a really simple question. and that is how do fluctuations in daily light integral actually affect the growth of, in this case, lettuce. And so the daily light integral refers to the total amount of light that plants receive in a day, and that's expressed in moles per meter squared or moles per meter squared per day. And it is widely used to make lighting recommendations for crops in controlled environment agriculture, especially for greenhouse crops. And there are algorithms available that allow you to control your supplemental lighting system to achieve that daily light integral inside of a greenhouse. But the simple question that we wanted to answer is, do plants actually need the same daily light integral every single day? Or if we have a sunny day one day and the plants get more light than what they would require based on daily light integral recommendations, can we take that excess light and basically apply it to the lighting requirement for the next day and then provide less light on the following day? And if we can do that, that would be a really simple way to lower lighting costs in commercial greenhouses. So we tested this idea with 2 different cultivars of lettuce, green salad bowl and red salad bowl, and they were grown under 6 different treatments where the plants either got a daily light integral of 15 moles every single day, or the daily light integral alternated between a high DLI one day followed by a low DLI the next day, with the magnitude of these fluctuations increasing as we go down the list here to pretty extreme fluctuations in daily light integral from 27.5 one day to 2.5 the next day. And so in the 2 data graphs that I have for this study, you will see the magnitude of the difference between the high DLI day and the low DLI day plotted on the x-axis. So in this graph, as we go from left to right, we have increasing variability in daily light integral from day to day. And we have the leaf area of the 2 cultivars right here. The 2 cultivars responded essentially the same. It looks like there may be a slight increase in leaf area with small fluctuations in daily light integral. And only when the daily light integral fluctuates very widely do we start to see a decrease in leaf area, and the results for dry weight look essentially the same, with only decreases in dry weight when these changes in daily light integral become very, very large. So from this, we conclude that lettuce can tolerate daily light integral fluctuations, and that we can carry over that excess light from a sunny day to a cloudy day. We're not absolutely sure yet, and we're doing more studies on exactly how much excess light can essentially be carried over to the next day. But at this point, we feel pretty comfortable saying that you can carry over at least 7.5 moles of light from a sunny day to the next day. And so I haven't shown a lot of particularly interesting science here, but what I really wanted to emphasize here is the practical implications of this, right? There's a really simple way to save energy, and it would be really easy to implement. And to see if this actually mattered, we developed a spreadsheet to allow us to do calculations to quantify the actual practical impact that this could have on lettuce production in greenhouses. And so in the spreadsheet, we used historical weather data from around the US, and we used that weather data to calculate, based on HPS lighting, the amount of energy that would be required in 1 year to provide supplemental lighting to lettuce grown in a typical greenhouse, in this case in Athens, Georgia. So here we have the total amount of energy that would be required, about 380 megawatt hours per acre in pink. And then blue shows the amount of energy that would be required if you actually can carry over that excess light from one day to the next. And we see, obviously, here that you use less energy for supplemental lighting, and this difference between these 2 different scenarios, we converted to a dollar number. And so what we found is that you can save roughly $7,200 per acre per year by implementing this, and that is for Athens, Georgia. I don't have data for College Station, Texas, but when we look at New York, Michigan, and Washington, we get similar magnitude financial savings that we can generate through this really simple method of adjusting how we provide supplemental light. In Arizona, the savings are a lot lower, and that is to be expected because, of course, in Arizona, you would provide very little supplemental light to begin with. And I can't imagine that any greenhouse lettuce grower in Arizona would actually have supplemental lighting because it wouldn't be cost effective in a climate like that. But I wanted to include this analysis here, especially for the graduate students in the audience, to give you an idea of how you can take the science that you're doing in your research and think about how you can translate the results that you get to an actual practical impact. When I say impact, I can see all of the faculty members who are part of the seminar cringing because we have to constantly justify the impact of everything that we do. But in some cases, it's relatively easy and actually pretty powerful to do that. And I think this is a good example of that. With that, I would just like to thank all of the organizations that have funded my research. In the past, that was the Fred C. Gluckner Foundation, the Georgia Research Alliance, seed funding from the University of Georgia. And currently, we have financial support from USDA, as well as the American Floral Endowment. And I wanted to include all of the people who have been part of the work that we do. But I think by now that there's a list of probably close to 50 undergraduates, graduate students, and other people, and it's just too many to all thank individually. With that, if there are any questions, I'll do my best to answer them. Thanks, Dan. Good question. Yes, so generally speaking, when you provide more far red light, you often do get thinner leaves. We don't see that always. And so we quantify that by measuring the specific leaf area, which is the size of the leaves divided by the dry weight. But generally speaking, yes, you do get thinner leaves. Only in extreme circumstances, I think. And so one thing that I want to emphasize when we talk about far red light, and I know that Xu Yang will appreciate this, for a long time in controlled environment agriculture, there has been this incredible fear of far red light because it induces stretching. So I would like to kindly remind everybody that sunlight contains about 18% of far red light, and plants seem to do okay when they grow under sunlight. So yes, there are limits to how much far red light you can provide, but we shouldn't be afraid of relatively small amounts of far red.
Mark, this is Bimu Patel. It's an excellent seminar and excellent work you are doing. I know you are probably not taking any measurements on chlorophyll A and B, if there is an impact on effect of this light on chlorophyll A and B, and also any other secondary metabolites. Does these lights changes those?
Yeah, so we've done some studies where we measure chlorophyll A and chlorophyll B, and so typically what happens with more far red is that the size of the light harvesting complex decreases, and that means that the ratio of chlorophyll A to chlorophyll B increases. And with regard to secondary metabolites, a picture that I showed early on that showed the lettuce that was grown in Eric Runkel's lab, you see decreased anthocyanin content with higher far red levels. And one thing that we're looking into right now is ways to induce high anthocyanin content just at the very end of a production cycle. because other data that we have actually shows that if you get anthocyanin in plants early on, anthocyanins will actually slow down the growth of plants because they absorb light that can then not be used for photosynthesis. So ideally, you would grow plants without anthocyanins and then do them near the end of the production cycle.
Thank you.
Hi, Mark. This is Murat. Wonderful presentation. It's always nice to listen from you. And as you explained, there are a lot of opportunities now we're seeing with the far red light and other spectrums, including UV, and really creating opportunities maybe for us to redefine the spectrum for the photosynthetically active radiation range. You explained that plants are responding to far red light with the leaf expansion, stretching, and more efficient use of the light. On the other hand, we also see response in terms of the reduced phytochemical or the secondary metabolites forming in some of these varieties and species. Do you suggest that far red light to be used in a more constant, fixed ratios all the way from the beginning of the transplanting stage to the harvest, versus a more dynamic, more strategic control of that, addition of that to the spectrum during the growth phase? I know this brings in the question of the cost for the fixture, designing and its implementation, but I wanted to hear your thoughts on that.
Yeah, so that's an interesting question that Neil Madsen at Cornell, or one of his students, actually can answer better than I can. So in our studies, we provide far red throughout the entire growing cycle, and that works really well. And I know that in Bruce Bugby's lab, they found similar things. One of Neil Madsen's students actually found that it makes sense to provide that far red during the seedling stage when light interception is most limiting. And so he did that, but then he doesn't necessarily see that the effects carry over if you move that plant from a far red rich environment to an environment without far red. And I actually had a long discussion about that. And what we think may be happening is that the plant acclimates to its light environment. And when you change that light environment all of a sudden from an environment with far red to one without far red, that plant may need to reacclimate to that new environment. And it may not be able to, at least initially, do very well as you move it from one environment to another. Anytime plants need to reacclimate to different conditions, I think you're potentially slowing down their growth.
All right, thank you.
Hello, this is Isabel Valles with the Potato Breeding Program. I would like to obtain your insights in terms of the type of LED lights for in vitro growth of plantlets under two different scenarios. One of them would be for rapid propagation, and then on the other hand, we want them to grow slowly for long-term storage.
That's an interesting question. So for rapid propagation, so intuitively you would simply say more light is better. But what we have seen actually with some colleagues in my department is that it's very easy to give tissue culture plants too much light, and we've had real damage as a result of that.
Actually, my experience with too much light with the LEDs is that they grow shorter, and for rapid propagation, we actually want them to grow taller so we can cut them and
I think then it depends on the light spectrum that you're using. You would probably ideally have a light that would have less blue and more far red in it. That's going to grow plants taller. Finding LEDs with far red may not be easy for reasons I've talked about. So one thing that you could try is to supplement the LEDs with incandescent bulbs, because they provide a lot of far red light. And I think you will see an increase in plant height. And then so for long-term storage, you would probably want to have a high blue LED that is going to keep those plants really compact. And no far red would be my guess. And obviously I'm just guessing here because I've not tried this.
Thank you.
Sure.
Hey, Mark, Matt Hardy here from University of Colorado. Is there a reason for the imaging section that you stick with the monochromatic measurements of the chlorophyll fluorescence? Have you tried just visual spectrum?
Yeah, I was going to talk about that and I took the slide out for the sake of time. So the beauty of doing chlorophyll fluorescence imaging is that leaf color doesn't matter. Right, we work a lot with plants that accumulate high amounts of anthocyanin. And if you take regular color images, image analysis actually becomes pretty tricky. It's easy for our eyes, but it's not as easy for a computer program to do it. So with the chlorophyll fluorescence, it's just looking at pixels above a certain brightness threshold. And so the system that I showed earlier, that's pretty expensive. But I now have an agricultural engineering student testing out a system using a Raspberry Pi with a camera. It's $200. It automatically analyzes all of the images, uploads everything to the cloud. So it's fully automated. It's incredibly simple and it's really cheap.
Yeah, I too was looking at the Raspberry Pi based imaging systems, and that's kind of why I was wondering if there was some benefit other than ease that I just wasn't seeing.
Yeah, so the color is one issue. It can become challenging.
Okay, thank you.
Kind of along those lines, I have a question about just how does the fluorescence imaging work systematically? Are you taking the image at the same time that you're exposing it to different types of light, or is there any kind of lag when you turn those lights off and then you take the image? And if so, is it, I don't know, microseconds? How does that work?
So what we typically have to do is we take the plants out of the growth chamber or greenhouse where we're growing them, and we put them in a light-proof grow tent with our homemade imaging systems. And then we turn on blue LEDs, and we take a picture when the LEDs are on with a black and white camera that has a filter in front of the lens that blocks all of that blue light. And so then the only thing that the camera can see is actually the fluorescence that is given off by the plants. There was actually a presentation on the ASHS website where I go into the principles behind this. And it's not our idea, but we really like it.
Thanks, super cool.
It's really simple. And so an interesting thing that we found, and this was another paper that Reeve published pretty recently, is that we can detect certain stresses really nicely based on the intensity of the fluorescence given off by the plant. So we did some work with atrazine, which inhibits electron transport at photosystem 2. And because electron transport is inhibited, cells that where atrazine has penetrated start fluorescing really brightly. And so we applied the atrazine to the root zone, and we can see the atrazine moving through the leaves. We have some really cool time-lapse images of that. Probably the coolest thing I've seen in my lab was to see the atrazine moving through those plants without any visible symptoms at all. Sorry, sometimes I get a little geeky about my research.
No, please do. That was exciting. Thanks for sharing.
If I didn't find the things that we do exciting, I would go find another job.
Are there any more
Nate,
questions?
by the way, so I see that Nate is on the call. So Nate is the student at Cornell, Nate Islands, who has done work with far red and transferring plants from an environment with far red light to one without far red light. And so he did not always get consistent results after he moved those plants. I'd just like to acknowledge him. Hey, Nate.
Hey, Mark, great seminar. Yes, we had a nice lengthy talk about photosystem stoichiometry and really started to make some guesses. And I talked about it recently in my A exam, actually, and we're talking about potential ways to actually prove that that postulate that we made.
Cool.
Mark, I think we have a question in the chat box. How do you think carryover excess DLI concept would work in fruit crops like tomato?
I think it would work in tomato too. Yeah, I would think that it would work in almost any crop. The exact amount of light that you might be able to carry over might be crop specific, but I would be surprised if the principle isn't widely applicable.
Thank you. Are there any more questions? If not, let's go ahead and thank Mark again for a great seminar. We're sorry you weren't here on the campus to share all of this with us, and we hope maybe next time you will be.